The project proposal was to develop a tool that would determine how E. coli manages protein synthesis stability. I started this project with the intention of designing a simulation that could be used as an alternative way to answer a scientific question without requiring a lab experiment. I created a simulation that would model the synthesis rate changes of robust and sensitive codons in E. coli under stress, specifically changing nutrient conditions while primarily focusing on the codon degeneracy lifting mechanism using the Python programming language in the VS Code IDE. This simulation included various computation techniques, including Monte Carlo simulations, Hill functions, Markov chains, and utilized GLMs and hierarchical models. First, let me discuss the research I conducted before starting development. I used these 6 Boolean strings as reference inputs for my research. Although all of them are slightly different from one another, some key themes include the strength and stability of proteins, the efficiency, dynamics, fidelity of translation, codon degeneracy and effects, translation in E. coli and other prokaryotic organisms, bacterial stress response, and the usage of computation techniques including stochastic simulation approaches. The databases and services I used were Google Scholar, Sci-Hub, Archive, and ChatGPT web search. Next, these are the articles that I referenced and used for this project. Instead of going through all of them in detail, I want to bring attention to some of the ones that I found to be the most helpful and that I used as a part of building this project, starting with Steps of Translation by LibreText Biology. I used this source mainly as a means to review the translation process and ensure that I would include several important components in the simulation. The main takeaway from this article was that release factors interact with consensus codons to terminate translation. I integrated this into the simulation framework by modeling the stepwise nature of translation, including ribosomal translocation and peptide bond formation, along with the completion of protein synthesis cycles. Next, global and gene-specific translation regulation in E. coli across different conditions was D. Jengs et al. And was published in PLOS Computational Biology. The article mainly concerns itself with the discrepancy between mRNA levels and protein levels and investigates the effect of various conditions on these levels. The first section I found useful was patterns of relative translation efficiency associated with codon usage, which stated that functionally relevant pathways share similar RTE patterns. This section also showed that a few pathways in E. coli exhibited large RTE variability under nutrient limitations, which has important implications for this project. The second section I found to be useful was codon usage partially predicts the cross-condition variability of RTE. In order to determine the effect codons had on RTE variants separate from the absolute value of RTE and mRNA levels, the researchers used a random forest model and found that there was indeed an undeniable correlation between codon frequency and cross-condition RTE variability. The next article that aided me greatly in the initialization of the project was Environmental Perturbations Lift the Degeneracy of the Genetic Code to Regulate Protein Levels in Bacteria by Subramanian, Palm and Kluzel. The main key takeaway I had from this article was that there is a codon hierarchy which categorizes codons as being either robust codons or sensitive codons based on their synthesis rate under amino acid limitation. The article also introduced me to the Codon Robustness Index, also known as CRI, which is a qualitative measure of codon robustness during amino acid limitation. I used this as a parameter to predict synthesis stability in the simulation. Furthermore, the article included a set of experimental data that I conceptually used to validate simulation outputs in the validation module. Now, in defining the precision and sequence determinants of protein synthesis rates, the article written by James Christopher Taggart, I realized that I can apply the principle of using boundary data to simulate transcription level dynamics, which is shown in the diagram to the right, on E. coli under nutrient and environmental stress. Another key takeaway from this article was that data on Shine Dalgarno strength, RNA secondary structures, and elongation kinetics can be used to refine translation rates of robust and sensitive codons. Another one was that the parameters for mRNA stability can be based on the role of RNase enzymes and secondary structures. Aside from utilizing those takeaways in the simulation, I also used the ribosome profiling data and RNA sequence data provided in the article as a conceptual benchmark for the simulation. There are 7 main components in this simulation. The primary component, the initialization module, sets up the simulation environment and defines all of the initial parameters and variables. Specifically, it defines the robust and sensitive codon categories, sets the initial nutrient conditions, and defines the biological components used in translation, such as ribosome tRNA pools and RNA degradation rates. The translation dynamics module simulates the protein synthesis process and incorporates the discrete translation steps. It uses Markov chains to model the transitions between the codon states during translation and incorporates Hill functions to account for the impact of nutrient concentrations on tRNA changing rates. The nutrient stress simulation module simulates environmental stresses and their effect on translation, primarily changes in efficiency. It uses Monte Carlo simulations to randomize nutrient depletion and replenishment cycles and dynamically adjusts rates based on nutrient availability. It also tracks changes in RTE for robust and sensitive codons. The mRNA stability and processing module simulates the impact of mRNA decay and isoform abundance on protein synthesis rates. It models RNA degradation using parameters set for RNase activity, incorporates transcript isoforms, and uses codon-specific rates and Shine Dalgarno strength to simulate initiation and elongation dynamics. Robustness and variability module simulates the codon hierarchy and quantifies the robustness and sensitivity of codons under various stress conditions. It calculates variance, Fano factor, and coefficient of variation for codon-specific RTE and integrates the codon robustness index, CRI, to determine stability. The validation and calibration module validates simulation results using experimental data and calibrates parameters. The simulated RTE variability is compared to experimental data to determine the accuracy of the simulation, and the model's parameters are adjusted using hierarchical models. The visualization and reporting module tracks protein synthesis rates, nutrient conditions, and codon RTE in real time. It generates graphs for variance, Fano factor, and CV, and also outputs a summary report at the end of the simulation. I have the full list of required libraries here, some of which can be substituted for one another, like Dash for Streamlight. This slide shows all of the program files for the project. The files in the first section consist of the major components that I just spoke about. The other 3, main, util, and input handler, are also parts of the program, so they're listed under other. As for calculated metrics, this project utilizes 4 distinct calculated metrics in assessing the evolution of codon variability and strength properties through nutrient and stress factors. Each metric takes together several characteristics which relate to a specific aspect of the functioning or the stability of a specific codon within the protein synthesis system. Variance is used to measure the spread of efficiencies around the mean. The Fano factor measures noise by normalizing variance with the mean. The coefficient of variation, or CV, measures relative variability as a percentage. And the codon robustness index, CRI, measures the strength or the amount of stress that codons can take, especially if nutrients are scarce. The simulation runs successfully with both custom user data and sample data implemented directly into the code. Although developing this simulation and debugging proved to be a difficult task, I had a lot of fun with this concept and would like to try to work on similar projects in the future.